Yeah, thanks for having me here and good to see all of you. Yeah, I became a yeast geneticist right away when I went to graduate school after exploring kind of a zoo of model organisms in undergrad and in rotations, trying out cell culture and zebrafish and Arabidopsis before finally deciding on yeast. And I've stayed there ever since. And my PhD is in genetics, and I'm a geneticist kind of by training and temperament, I'd say. And so when I had the great fortune to land at UW in 2008, it was a really fun time because it was the same department as Stan Fields and Doug Fowler were working out some deep mutational scanning ideas, and Jay Shenduri and his lab were also doing some saturation mutagenesis ideas. And so I was kind of an early adopter of mutational scanning. And we started applying these methods to all sorts of the things that we were already interested in for other reasons. And so it was really a natural progression. I think geneticists are by nature greedy. So this idea that we could make all the mutations and measure all of them at once was very appealing to me. And so um, I've been doing things in the kind of basic science realm of trying to understand how genomes evolve and how mutations have consequences. And more recently have been realizing that the same types of methods apply to any gene that you can get to work in yeast. And so there's a whole, battery of human genes that you can craft assays for in yeast. And so we started to turn some of our tools to those. And that's been both a real learning curve, but also great fun and very satisfying to get involved in. And oh, Laura, you asked about the origins of the Deep Mutational Scanning Symposium and what has turned into the meeting that I'm very excited about going to in a few months. So this really came out of two people. One, Elisa Wong, who is a postdoc in my lab, who is also working with Doug, and also Olivia Kosterlitz, who is a graduate student at the time in Ben Kerr's lab and continues to work with Ben actually on some things. And I was on her thesis committee. And she was trying to apply some deep mutational scanning ideas to some evolutionary biology questions, which I thought was pretty interesting, and was having, some of the technical challenges. And, in her committee meeting, I was like, you just need to talk to the people in my lab, and then, we can have, this interchange of ideas and trade tips and protocols and everything. And I was like, here, let me introduce you to Elisa. And then what was going to be kind of a glorified joint lab meeting turned into this like I think we had almost 100 people there, and they ended up getting like a company to sponsor part of it and everything. And it just really showed that there was this desire for practitioners in this field, even though people were applying the technology to all sorts of different genes and all sorts of different problems, that there was a unity of thinking and a style of data analysis, for example, and experimental design that kind of could bring people together and where people could really learn from each other. And so starting with Elisa and Olivia, that kicked it off, and it's been really satisfying to kind of watch it grow and get bring in more people every year from its little Seattle roots here. So I'm excited to get to go again.
No, that's very cool. Thank you for sharing that. I hadn't realized quite how organic it was. Um,
Yeah.
and
And
yeah, what would you say is like the biggest surprise then, fast forwarding kind of 9 years? What's what in terms of how the field's developed?
Oh, yeah, I think it's just incredible how big it's gotten. And so part of that, of course, is the Atlas for Variant Effects Alliance, which it's gone from kind of a ragtag group of people collaborating and trading ideas to a field in its own right and a community of practice, right? And so I think that's been really exciting to watch. And also, I mean, it's not surprising, but it's gratifying to see how creative everyone is. It's really interesting just to see how many people come up with interesting ideas, both on the technology side of things and the assay development side of things, but also in terms of like, what can we do with these data? How's that going? And so, the move to big machine learning models and everything and how these data feed into that and the ecosystem back and forth, I think has been really fun to watch develop. And I'd love to hear more from you about how the Ave Alliance developed and where you think that's headed in the next few years.
Yeah, sure. So, well, I guess I've been lucky enough in my career to kind of get involved in some big international consortium projects, and I arrived at Sanger in 2003, as Laura mentioned, and that was at the very back end of the Human Genome Project, and so seeing that kind of collective effort And then through various other projects like the HapMap project and the Thousand Genomes project, but then also being on the sidelines, seeing things like the Human Cell Atlas come together. And it seemed that there were certain types of projects where that really worked well. When there was a kind of a real shared mission across a bunch of different centers, and there was the opportunity to generate a data resource that would kind of outlive the people and expand and value beyond the people who were generating it. And that combination of kind of culture and mission, I think, really kind of resonated with me when I first came to the kind of the 2020, which was the third symposium, I think, wasn't it, in Seattle? And that really struck me that this felt like that kind of problem where there was that kind of community with that kind of culture, albeit in a very nascent form, and the kind of problem, the mission that was really important. That there was clearly the right answer is to determine the effects of all variants in the human genome, and clearly that can't be done by any one group in isolation. And it requires the combination of large-scale data generation, the data coordination and the infrastructure, all the kinds of things that those other projects required and that the other projects showed could be done. So it felt very much like this is the next big challenge, in some sense, the kind of the next big frontier, the ultimate frontier, to go from a human genome sequence to then knowing what every single possible variant in the genome does. So that was really, I think, the genesis of it, and I think that's kind of started to bear fruit, in really kind of unexpected ways, as you say, because the community is so kind of varied and creative. And for me, I think the attractive things have been the interdisciplinarity of it. And so, me as a rare disease geneticist, once evolutionary geneticist, kind of coming, talking with protein biochemists and hearing terms that I'd long forgotten back in my undergraduate biochemistry days and struggling to remember what they meant. And so that was really invigorating, I think, having that kind of interdisciplinarity, but also the kind of the youth of the group. I think I've been fortunate to be part of different collaborative communities, and this, I think, is probably the youngest and most vibrant because of it. So that was really attractive, and also just the fact that there's a very obvious real world impact that feels tangible, that we can kind of collectively work towards. So it's not so much a kind of build it and they will come, so much as everyone wants this yesterday.
Yeah, I remember having a conversation with an interviewing graduate student who I won't name in case he wouldn't like me to, but he was trained as a genetic counselor and was applying to our PhD program. And I was like, what do you think? If you told some parent that the yeast told them that their baby was at risk of this disease, like what would they think of that? Would they believe you? And he was like, well, it can't be worse than what we're already doing. And so it's been really amazing to me to watch some of our yeast results from an assay do have clinical relevance. I had someone reach out who one of the genes we've been working on lately is G6PD, glucose 6-phosphate dehydrogenase. And we had a hematologist from the Hutch say, hey, I just found this variant in a patient of mine, and I think it's causative. Is it in your data set? And we looked it up, and there it is. It looks like it's an actual, uh, loss of function mutation. And that back and forth is incredible to me. And just the real-time nature of it too. And that it was that, the idea that we have an atlas and that somebody comes to us and says, hey, is this in your atlas? And you go, oh, yep, there it is. Like that has actually come true. And, even for a lab like mine that is really a yeast lab, we've been able to have that kind of impact. And so watching the whole field, tackle the whole human genome like this is just incredible. So, you've done so much work with rare disease and things. How do you see the kind of understanding the kind of nature of disease, the kind of what are the fundamental insights that you think we've gotten from this new kind of comprehensive way of approaching it that are beyond just having an atlas you can look things up in?
Yeah, no, I think it's a really good question, but I think we're very much in our infancy because I think we're scratching at the surface of what's possible with these maps. I can absolutely think of a future in which any drug target that's being
Mm.
used by any pharmaceutical company, they'll want to have a variant effect map or several variant effect maps of that drug target because it will be so useful at different stages of drug development. Not just do these individuals really have this disease, but actually what are the different mechanisms that happen, that are possible within the gene, and which ones are tractable by which kind of therapeutic
Mm.
approach. So I think we're just scratching the surface at the moment, and we're kind of it feels like one of those data resources we're using currently for the reasons that we intended, but in 5 years' time we'll be using for reasons that we didn't intend, and those may well
Yeah.
actually be more impactful than the original ideas. So yeah, so you mentioned about a little bit about your yeast genetics. And I was wondering what you kind of felt that the yeast could kind of uniquely bring to the kind of the Mave community.
Oh yeah, I'm still a big fan of model organisms, despite how incredible the field has developed to be able to do many kind of yeast-defying the world, right? CRISPR has allowed lots of things to become more like our system and all the ease and speed that we're able to do things with. Um, I think that yeast is still a great assay tool and fast and easy, and also great for training. So when I was teaching at the Cold Spring Harbor yeast course, for example, we did a deep mutational scanning lab as part of the 3 weeks that we were there, and so that's pretty hard to pull off, I think, with a lot of other systems. Um, and still had its challenges, by the way. I'm very excited to see similar courses going forward at other places now with learning some of the basics and getting people together and how to propagate some of these ideas. But the other thing I think is fun is that, yeast is not just a model, it's like an organism that's used for things. Like big fermenters that manufacture drugs and small molecules and all sorts of things. And a lot of directed evolution work that is kind of a precursor to this field, I'd say, has been applied to those types of engineering problems. And I haven't seen as much melding of those as I think would be interesting to do. Maybe some of that's hidden in the companies. But that idea that the yeast can be both a model but also an organism in its own right that does things, I think is one thing that keeps me going back to it, um, for sure.
Oh, that's interesting. What would you love to get yeast able to do that it can't currently
Ah,
do?
oh, that's a fun one. Oh yeah, making things better is a hard one for sure. We've been working on brewing yeast some and thinking about assays for flavor and how you can multiplex things that are, how do you make a cell autonomous assay for something as vague as flavor that, it does reduce to chemistry to some degree. But yeah, it's fun to think about. We had happy hour in my department the other day where we put up on the board like what would you like us to make our next super yeast to do, because we've been doing some evolution experiments to make yeast do various tricks, and so people did come up with some creative ideas, so.
Oh, excellent. No, I hadn't thought of that at all. But yes,
Yeah,
is there anything
what
like
systems do you mainly work in, and why do you how has that changed over the years? I mean, going from a more history perspective, you've had to kind of choose your way to what makes the most sense.
well, I'm definitely a human
Yeah,
geneticist.
of course.
With a very brief foray into sweet potato
Hmm.
genetics, just because that turns out is particularly fascinating for Polynesian prehistory. Um, but
Love it.
otherwise, we've done a little bit of mouse work, making mouse models of neurodevelopmental conditions, but not in a mutation scanning context. But for mutation scanning, we'd be really focused on HAP1 as this amazing cell line that kind of shouldn't really exist and shouldn't really work, but does.
And
And
again,
that's
kind of
been
yeastifies human cell culture.
it
Haploids
absolutely it
make
does,
things
and
easier.
it
Yeah.
turns things haploid again, which bizarrely I was at one of the few, because my PhD was on the Y chromosome, and the Y chromosome is constitutively haploid. I was like the only human geneticist that kind of used to haploid world are well Y chromosome geneticists, but so it was initially confusing to me when I moved out of the Y chromosome to the rest of the genome that there were two copies of everything, but I managed to cope in the end. But so yeah, so HAP1 feels like a bit of a return to home, but it's so incredibly useful. It has such high HDR editing rates that it becomes a really tractable model, and I don't think we fully understand why it's such a tractable model. It's just one of those serendipitous kind of biological things. We could have ended up with a haploid cell line that was incredibly bad at homology directed repair, but fortunately we didn't. So
What do
yes, so
you think
haploid.
are the kind of biggest technology challenges that remain to be solved by the field before, we can go for everything? Like what would you like to see be easier in your lab?
Yeah, I think there's a kind of I think what I see is a twin track approach. I see us kind of working to scale the things that we can scale at the moment. So I think for those, the scaling is the big challenge.
Hmm.
How do we scale data generation? How do we move from, one gene, one project to 10 genes, one project, 100 genes, one project? For me, the bit that feels least scalable is the kind of the cell culture work.
Yeah.
And that's the bit where I think a real investment by the community, the more broader community, in scaling cell culture using laboratory automation methods of the future. I think that will have a huge impact on all kinds of cell-based screening, not just MAVES. So I think that feels like something that funding agencies should really think about how they incentivize. Um,
And that's
and
funny
I,
for me to think about for yeast because growing the yeast, not our main problem.
yeah, But so yeah, scaling the things that we so in HAP1 we know that there's of the order of 2,000 essential genes that we could essentially apply the same assay
Yeah.
to. Several hundred of them are known disease genes, so we have an immediate target list there. But actually our ability to run those experiments is limited by scaling. So that's fundamentally the limiter there. But then also we've got the kind of the fact that we don't have assays for many of the genes that we'd want to have assays for. And so I see a twin track of kind of technology development on assays at the same time as scaling ones that we know work. So I
Right.
think
Yeah.
that,
The,
and that's
the
why
boutique
it's not an
approach
either
to
or.
assay development is both an opportunity and a challenge. And when you can create kind of generic assays that you can just scale by brute force in some ways versus when you need that. And I do like in some ways having multiple assays per gene too. I think that's another direction I'm excited about things going in where, function doesn't just have one definition and things are putting things more in contexts that matter, both genetic context and environmental context. I'm excited about where that I see some of that going.
Yeah, if one thought what would the ultimate experiment be, then you'd kind of really like to know about every single variant in the human genome in every single context
Right.
with a kind of single cell multi-omic readout. So to give you the best chance of understanding function. I think that's probably a much more multidimensional functional kind of readouts, like you say, and ideally you'd want to know the entire molecular state of that cell. This is very far in the future, obviously, and not things we can do, but we can make steps there. We can potentially make single cell RNA-seq maps of every variant in particular genes. That can start getting into the multidimensional space, or we can use the kind of cell painting methods and the more imaging based approaches.
Sure.
So I think we're starting to move into that multidimensional functional space, but we're not yet at the stage of being able to do the ultimate experiment, if one can define what that is. I don't know. Would you have a different idea of what the ultimate experiment might be?
Oh yeah, well, I mean, I'm really interested in the genetic compatibilities, like how, different genetic context and epistasis, and will we ever have a true like generalizable model of epistasis and when genetic interactions matter and when they don't? That I'm very curious to see how that develops in the next few years because the combinatorial just blows up too fast, right? And so we kind of can't get there from here with some of the types of genetic interaction questions that I'm particularly interested in, but that really matter for the generalizability of a lot of these scans. So getting more data on different haplotypes or different combinations or doing that on purpose and seeing how can you go beyond pairwise is really interesting. One of the things that I've really loved learning about as I get more into human genetics is that, a lot of the fundamental evolutionary biology questions are the same. Genetic variation is the same problem everywhere. And there are these fascinating genes that have tons of genetic variation and really interesting evolutionary histories. And so that while we get the clinical relevance is like a great bonus in addition to these basic science questions that I've always been interested in as well.
Yeah, I think it's interesting being having a yeast geneticist and a human geneticist here because obviously the evidence of genetics interactions in model organisms like yeast is so abundant and so strong, and yet the evidence of disease relevant
Right.
interactions in humans is so weak, And is that because we don't have the means of searching for them in humans, or is that because it doesn't happen so much in humans? And I think that's a big conundrum for the whole field. I do think one of my kind of population genetics past is humans are a young species. We don't have much genetic variation. And so the likelihood that you have genetic variants that interact with one another, and don't have a big fitness effect on them individually, is quite low. So it might be that epigenetics exists in theory for humans, or those kind of sort of epistatics or interactions. It exists in humans, but we don't see it very often. So certainly in our genetic studies, when
Yeah.
we've done whole genome or whole exome sequencing, we've searched for these kind of unusual epistatic effects and really not found them. And quite a few other people have and in
Right.
different ways. There are a few nice examples, so where we know it definitely happens. And one of my favorite ones is around the thalassemias actually, and and these interaction between sickle cell alleles and alpha globin deletions
Yeah.
and things like that. So, but that's where you've had kind of a strong selection pressure creating those variants in the same geographical location. So
Well, and I think that highlights some of the kind of. I wanted to talk to you a little bit more about the clinical implementation of this because it's so often there's a kind of a like we know this that oh well is there great evidence of this? No, we probably don't have to worry about it. And so when you see like an obvious loss of function variant next to a variant that doesn't do anything, you're like well that haplotype you should say is bad because it's got a bad mutation on it. And so but in the clinic, they're like, well, no, we don't actually have a test for this exact haplotype, so we're going to say we can't say anything about it, which is sometimes frustrating. And so, what do you see as like the kind of connections that need to be made for the biggest clinical relevance in terms of actual implementation of this and getting like how do we make doctors and genetic counselors understand these data well enough to be able to use it?
yeah, to be honest, I'm actually really optimistic about this because if you think of all the challenges that could exist, then actually most of them we're part way there on, and we can kind of see where the future goes. So, they're scaling the data generation, which I think is the biggest challenge. Then you've got scaling the data analysis. And actually, compared to some of the other projects that I talked about, like the Human Genome Project, where really the data analysis completely didn't scale and one had to invent completely new methods, actually the data analysis does scale, I think, for in MAVES. The data infrastructure that you need to bring all these collective efforts together, we have that with MaveDB. We've got the kind of the benchmarking tools and benchmarking kind of philosophies to enable us to say what data is good data or good enough data. We've got the kind of first pass of the clinical guidelines that say how these data could be used, with the ACMG guidelines and
Hmm.
so all of these barriers that in theory you could imagine exist, actually we're part way to solutions for. And you can even imagine how we could get MAVE data in the hands of clinicians and diagnosticians at the moment they're making diagnoses, because they're all using variant effect tools, whether it's decision support tools or variant effect predictors. And so there's already a digital route through to those clinical eyeballs who are making those judgments. And for example, there's MAVE data that can be seen in the Decipher portal, which is a kind of free to use clinical decision support system. So actually, in my experience, the clinicians are waiting for these data.
Yeah.
They know this can come. They have these problems. They know their problems. So you don't have to motivate them with this as a problem that needs to be solved. They're super motivated by that. And I think we've got enough test cases now where they're convinced with like the BRCA1 data especially,
Sure.
that actually this can be really powerful. And ClinVar has essentially gathered over the years the benchmarking data that now
Right.
enables to say whatever experimental system it is, whether it's HAP1 or yeast or indeed cell free, we've got the equivalent of model organisms, except in the form of hundreds of humans, carry some who have clinical manifestations and others in large population cohorts who don't. We through our previous work, we know of hundreds of humans carrying the variants that we've now just profiled in whatever system we've used. So I feel we're in a really good place. We just need to turn the dial up on most of them, and the hardest one is the scaling of the data generation.
Right.
So that's my take on it. But I don't. Do you have a different take on it with yeast? Maybe it sounds like you're not so worried about the scaling
Oh,
of
well,
data
I'm not
generation.
as involved in that part of the, although I've trained several genetic counselors in my lab, so I feel like, the supply chain, we're good at that part. No. No, but it is exciting to see. It's a really fun and cool time to be involved in all this. Oh.
Yeah, is there going back to how the field's developed, because you've got this longer time frame than I have on it. Is there anything that you thought should have happened or would have happened or you hoped that had happened that hasn't?
Oh. Um, one of the reasons for the first symposium was because evolutionary biologists like Olivia and Ben were getting more into the field, and I would love to see that. And so I feel like the evolutionary biology field could have picked it up a little bit more. This idea of like one thing that really I love about these saturation mutagenesis methods is the idea of what can happen versus what does happen. So, my lab, we do a lot of experimental evolution, and so we let the yeast do what they will and then sequence them at the end after they've been through some selective regime and then try to understand what does this have to do with anything. And so a good example of that is that we kept seeing gene amplifications of this one particular transporter for a nutrient. And if you're limited for the nutrient, increasing the transporter, great, makes sense. And I would get questions all the time of like, why don't you ever see increases in expression of that gene instead? Do you get promoter mutations? Do you get transcription factor mutations? And so with Matt Rich and Stan's Fields Lab, we ended up just one day being like, fine, let's make all possible mutations in the promoter of this gene and see like, are any of them or any combination of them as good as what you can get by just amplifying the gene by an extra copy? And so we were able to answer the question definitively, no. Like the
Right.
fitness benefit is just not as great. And so that like was really satisfying and allowed us to kind of explore evolution in a way that kind of was harder to do by having to just take what evolution gives you. You could start it off with some things and then say, okay, now what do we get? And I think there's several people absolutely working with that mindset in the field that, and I want to see kind of more of that from the basic scientists and the evolutionary geneticists. How can we use these methods for real basic science questions about genetic variation and evolution?
Yeah, that's interesting because we're also kind of going through an explosion of data in terms of reference genomes for other
Yeah.
species. So this should be, the next 10 years, it'll be, the 10-year biggest decade in decoding 4 billion years of life. This is when reference genomes will be available for tens of thousands, hundreds of thousands, up to a million species.
Well, and
Um,
that's one thing that we've been trying to go back and forth between is like, can we use like single missense mutations to interpret the more complicated alleles that we see from natural variation when we go and look in the world at different yeast strains or species even. So what translates and what doesn't. So
Yeah, exactly. So we'll have there's two explosions of variation data, one
mhm.
in kind of in model systems and one in organisms, and I think comparing the two is going to be a real rich vein for evolutionary biology. Are there other fields that you think could adopt the mutation scanning more? Because I'm thinking of especially of the keeping the kind of protein structure function kind of people really engaged in the
Oh
community.
yeah, I mean it's been incredible for protein exploration, I think. And I think all the modeling stuff that we can design stuff to spec now is incredible. And so how the interplay between doing an experiment and doing prediction or an analysis that goes back and forth, and when will the experiments be less needed? That's a moving target. So that's going to be fun to watch for sure, particularly being at UW with David Baker up the hill.
Mm.
I do think some engineering strategies, and one thing that I'm particularly interested in is weird mutations. Like how do we understand dominance? How do we understand gain of function mutations? I think loss of function mutations we're getting pretty darn good at recognizing the features of, but some of these weirder ones, those are a little harder. And so I'm getting more interested in some of those and what we can we design assays to find them? Can we get there with just single, or is that one where we're going to need bigger mutagenesis or different kinds of strategies? So borrowing some ideas from directed evolution. And because a lot of times directed evolution, they're like, here's the one that did the best, so we're going to take that and then now go mutagenize the heck out of it again and then do steps like that. But, how do you learn from everything else and all the mediocre ones in the process, I think, as still an open question in a lot of ways.
Yeah, no, that's it takes me back because actually my undergraduate dissertation was on directed evolution, and so there was obviously an interest there from many years ago. But yeah, that is fascinating. I do think there's a huge opportunity in keeping the interdisciplinarity of the field and not necessarily getting too clustered around one particular application. I think we need to kind of we need to kind of nurture the community in a way that we kind of maintain interdisciplinarity because it's such a classic scientific kind of direction of travel to kind of go with your tribe and only speak to people that speak the same language, and it really holds us back, I think.
And that's one thing I really like about the symposium is that it does bring people together who are, it's rare to have a symposium organized around a method almost, right? It's like I go to like a meeting about yeast or a meeting about evolutionary genetics or a meeting about a biological process like chromosome integrity or something. And to have a meeting that's more focused around like a strategy of thinking about science is I really, but it does lend itself to a diversity of systems, approaches, applications that I think is really interesting and fun and a little bit
Mm.
different from the other kinds of meetings that I go to.
Yeah, no, exactly. That's I guess it's one of the things for young scientists listening is kind of don't go to the same kind of meeting. Try and find different kinds of meetings. So yeah, we'll try and be at the intersection of different tribes.
Yeah, absolutely. One thing that I can't help but notice is that both of us are also interested in copy number variants. So how did two people who work on copy number variants get into this field where we're now obsessed with single point mutations?
Yeah, no, it's interesting. I could say a little bit about my journey at least, which is I was just my PhD I mentioned was on the Y chromosomal markers, and actually bizarrely copy number variation was like the one thing I wasn't looking at because I was looking at SNPs, microsatellites, and mini satellites because they were the most variable markers. So I've always been interested in lots of different types of variation. I think what really struck home with me was that when the genome came out and suddenly there was the potential to look around the genome at all the variation, but we didn't have the technology. We didn't have the technology to do the sequencing. The next generation sequencing wasn't there. But what was there was this ability to use microarrays and to look at large scale copy number variation genome wide. So that ability to discover genetic variation genome wide came first for copy number variation. So it was more that the genome perspective was kind of, and that kind of openness to looking at different types of variation was there, but the technology that came along first was the copy number variation
That's
one.
so funny because I've got nearly the same origin story. Yeah, ye old microarray days, that was a fun time. I was yeah, during that, and also for me it was partially that you go where the yeast take you, and so we had these evolved strains of yeast that had adapted, and it turned out one of the methods they used was chromosome rearrangements and gene amplifications, and so it was either like ignore that or get on board with what the yeast were interested in. So but I think it also comes from just our perspective as geneticists that we want to know all the variation, understand all of it, and that it makes a natural approach to then be like, oh, we can saturation mutate this whole thing and get all of it. Yeah, no, that's kind of the similar mindset, I think, in terms of like, let's understand everything that goes into a genome.
Yes, yeah, yeah. I think that goes back to your comment about being greedy as geneticists, doesn't it? Yeah, yeah. No, I see what you mean now. Yeah, I know. I did think it was very interesting at the time. Going back to the point you made around yeast and can you replicate with promoter mutations the effect of a duplication or gene amplification? One of the things that struck me some of those maybe 10, 15 years ago when people were starting to map a lot of the loci in domesticated species, especially domesticated animals
Yeah.
that were at the some of the key traits that we'd selected for, that many of those turned out to be copy number variations.
Hmm.
And I don't know whether anyone's done any analysis to say if a disproportionate number, but it seemed that there was a disproportionate number. And I do wonder whether that relates to some of those trait-related kind of changes being very hard to replicate at a sequence level what you're doing at a structural level.
Yeah, and I think it's fun to see people also taking kind of similar high throughput approaches to like making lots of copy number changes or making lots of chromosome rearrangements and trying to understand in a similar mindset as deep mutational scanning, kind of the all the things. Huh.
Yeah, exactly. And I do like the technologies that allow the exploration of that broader space. One of the things we've been doing recently, that this is work done by Vanessa, who hopefully is going to be presenting at the MSS, is one of my graduate students, and she's been putting in things like G quadruplexes
Oh
and
yeah.
stem loop structures into 5' UTRs. And so that again comes from that sense of what's the what can we look at everything? Let's not just look at the single base changes. Let's look at the insertions, deletions. Let's look at the kind of weird, unusual DNA structure forming sequences that we know exist. There's fascinating enrichments of G quadruplexes in promoter sequences, for example.
Yeah,
Yeah,
very
so yeah,
cool.
so I especially like those technologies because a lot of the technologies that we have for generating the variation that we use in these MAVs can be used for broader classes of variants than just SNPs. And so I definitely encourage people to really not just look at the single base changes.
Yeah, absolutely. Now we've been doing some transpose on mutagenesis screens too, so which are again classic. I love the idea of like these classic genetic methods. How can we modernize and genomicize them and take advantage of all of our modern tools, but keep the rigor and the thought process behind kind of these classic methods. It does make you really appreciate the foundational people in the field too, who kind of had to do it all with lesser tools though.
Yeah, and it's interesting to think how will the graduate students of today's work be revolutionized in 20, 30 years time?
Yeah,
What's the thing that seems
absolutely.
completely impossible today that'll be, completely routine in 20, 30 years? That's the speed at which all of us are kind of the whole field's moving means that all of us will kind of start our careers doing things that look completely trivial, halfway through our careers.
I remember one of our very first projects for this was looking at replication origins. And in one experiment, we recreated somebody's figure that took all sorts of hard labor and that was a bunch of, plus or minus for whether the mutation worked or not. And here we had this beautiful quantitative data for the whole sequence. And I remember being like, wow, okay, we're onto something here. This is going to be fun. Well, maybe we have talked enough and we should turn it over to the audience here to ask us some questions. I've really enjoyed this conversation though, Matt.
Yeah, no, that's been great. Yeah, I agree. Let's turn it over and see what questions people have for us.
Thank you, Maitreya and Matt. So we have a few questions in the Q&A, and I guess I'll go through them one by one. So the audience members, if you would like to ask a question live, please feel free to raise your hand, and then we'll make that happen. So the first question we have is for Matt. So can you talk more about your approach to assay development for non-HAP1 essential genes?
Yeah, so I think in terms of my lab, we're kind of looking at beyond the essential genes to what can we learn from these single cell kind of multi-omic assays, especially with single cell RNA sequencing. And there's been some interesting work done on this by the Illumina team and others subsequently. It's not a cheap process, and so scaling through has cost implications. But that kind of that approach of let's try and be much more agnostic as to the different possible functional, types of mechanism that can exist in any one gene. So, we know, for example, just from our clinical genetics, that there are some genes, and filament A is a great example, where there's, I think, at least 7 different diseases that can be caused by different types of mutations in filament A because of different mechanisms in different parts of that protein. So we already know, actually, a very high proportion of OMIM genes have more than one disease associated with them. So it's not so much the exception as the norm. So I think it's really important we do have these methods that aren't just looking at loss of function, and aren't just looking at HAP1. At the moment, we're kind of focusing on HAP1. We think the value that we at Sanger bring is is kind of scaling up some of these technologies, more so than developing wholly new technologies. So there's lots of great groups developing new technologies, new specific assays for specific genes. So we're trying to focus on the areas where we can add the most value by scaling up assays, and HAP1 kind of lends itself to that. I think once you move into iPSCs, for example, the costs of culturing go up dramatically, as well as some of the technical variability. So that's just what our group's kind of thinking about in terms of going beyond essentiality. But that's also very much informed by our sense of what we bring to this community, and focusing on the things that we can do that add the most value.
Thank you for the thoughtful response. So the next question is for the both of you. So do either of you see a need or possibility of MAVES in diploid human cells?
I don't see why not. I think
Yeah,
the idea is we've thought about doing things in diploids in yeast as a way of, looking at dominance, for example. And so I think you can get at different questions.
yeah. There are some human diseases where you only get the disease in the heterozygous state and not the homozygous state. And so you actually need diploidy to be able to model those diseases. So dominant negative is a good example where you've got one mutant allele and one wild type allele. So I think there's absolutely necessary that we do move these assays to diploid human cells, not just because there's mechanisms where diploid is important, but also to get that broader set of cellular contexts. We're not going to have haploid cells for all cell contexts that we'd like to have.
Thank you. And third question, Matt mentioned the growth in available reference sequences. Do you see a use of pan-genome references in any step of the MAVE process, from design to analysis/interpretation?
Oh, that's interesting. So I think some people mean different things with pan-genome references. I do think from a disease perspective, you can get very focused on humans, and you can get very focused on human variation. I do think it's interesting to think about, but given the flexibility we have for what alleles we introduce, we could absolutely be introducing more complex alleles that mimic things that are seen in other species. And we may well learn more from doing that kind of approach rather than just exploring one-step mutational changes from human reference sequence. So I think from that perspective, from a multi-species perspective, I think there's value in thinking about that. From a kind of even within humans, I think it's important that we are generating maps that are useful across all of humanity, and I think we need to understand where that falls down and where we might need to do experiments differently to try and counteract that. In general, I'm optimistic that the kind of maps that we're generating now are going to have very broad cross ancestry utility, because they're typically looking at very fundamental protein, how proteins are produced, how proteins are stable, and I don't see that changing very dramatically, at least in the coding space. I think in the non-coding space there might be more variation, but that also goes back to the fact that humans are a very young species. We don't have much variation as a species, despite the fact that we all look and show up very differently to each other. I don't know, Maitreya, did you have anything you wanted to kind of add on that?
Oh, not particularly. Yeah, interesting to hear you reflect on it.
Yeah, I guess one of the things that we do see, and we have seen in some of our studies, just purely from the genetics, is it's not a level playing field in terms of diagnosis. It is harder to diagnose, especially people with African ancestry, because the reference data sets that we use in terms of population variation aren't as rich. And arguably they'd need to be richer because there's greater diversity in Africa, because that's where the origin of humans is. So we know that the existing tools that we're using to diagnose are biased, and I would argue probably more biased than our tools that we're developing with MAVES. So I think MAVES can help actually to correct that bias, and I'm really I'm really kind of keen that and I bet it may well be a twin track approach for correcting that bias in diagnostic tractability by both kind of increasing reference data sets of population variation in those populations that have been underrepresented, and also scaling up the MAVES. So I'd see both being important.
Awesome. So a kind of sort of a follow-up question to that question is that when do you sort of expect genomic data sets that account for ancestry or ethnic background to become clinically useful, and what might their adoption in routine care look like?
So I think the existing tools that we have are useful across. So for example, when we look in our DDD cohort, which is a cohort of 13,000 families with developmental conditions, we see that we have the same diagnostic yield in parent-offspring trios with African ancestry as with European ancestry. Where we see the gap is when we don't have both parents available and the child. And if we've only got DNA from the child, that's where we see a lower diagnostic yield in individuals of African ancestry. So the existing tools are useful. And by existing tools, I mean in this context, the broader diagnostic tools that we use in routine clinical diagnostics, not just the MAVES. But I definitely see the MAVES being advantageous in terms of leveling the playing field in terms of diagnostic yield, especially when we don't have the parents available. That's just what we've learned from developmental disorders. That's because we have a high proportion of dominant conditions caused by de novo mutations in developmental disorders. It can be a different pattern in different parts of the world depending on their demography.
And I think there will be increasing data sets that explicitly test this, that create the same, a library on different haplotype backgrounds and say, like, did it matter? And so working in pharmacogenes, there's so much variation in these genes that they tend to have more variation than your average gene. And so there's more of this going on. And it's certainly something we've thought about doing, of saying, can we introduce a common haplotype from a different part of the, different parts of the world, different parts of human ancestry that's common in other areas that, onto the library and say, like, does that matter? Does everything shift up and down, or are there, epistatic effects that are non-additive that are interesting that we need to know about, or what? So I think there will be more data to bring to bear on this as well coming. And some of the people, including some people I notice on the call, are contributing to these types of data sets.
I think one thing that's also worth mentioning is that, which is sometimes underappreciated, is the human genome reference that we're currently using is a composite of many different individuals. And the biggest single contributor, which I think accounts for the majority, is actually an African-American individual. So some people, I think, have the impression that it's a European genome, the reference, but it's actually more African-American than it is European. And African Americans are typically an admixed population of majority African ancestry and minority European ancestry.
Good. We have more questions in the chat. I think Priyanka will take over from here.
Yeah. Hi. Yeah. So the next question is that how do you combine the utility of MAVE assays with protein structure modeling and de novo protein design that are becoming more common?
Oh, I think this is definitely a Seattle question.
Yeah, no, it's been very exciting to be on the fringe of this emerging area, because it's just incredible what you can do in protein design these days, and that these mutagenesis strategies are so useful in elaborating designs and testing them has been really fun to watch. So I think they are learning from the data sets where you can more carefully understand the consequences of mutations and incorporate that into the models. But you can also take a model and then see, like, can we make it better by making mutations that maybe they're not so good at predicting. So I think those are both really exciting directions, both to feed back to the models and to improve upon them, and to see if you can get proteins to do more things that you're interested in. One thing that I find super fascinating is designing proteins that don't really look like natural proteins at all, but still do things. And so I think figuring out, like, are all the rules similar in those contexts, or do we, have stuff to learn about protein structure function from these really interesting new kind of designed proteins that we can kind of extract out through classic mutagenesis methods? I guess we'll see.
Great. Yeah, thank you for the discussion. The next question is, where do you stand on the need to repeat MEVs in more disease-relevant cell types than, for example, HAP1, would require a doubling or more of workload, but may yield different or more accurate results?
Yeah, so I could say where essentially it's an empirical question for me, because we have really good benchmarking data. So if you have 200 individuals who have a disease and they have a pathogenic variant in a gene, and you've got 500 individuals who don't have that disease but do have a different variant, different variants in the gene, then you've effectively got 700 model organisms, that happen to be humans in this regard. So the question then is, how well do the data, whatever the origin of the data is, how well do they predict, what are the 200 and what are the 500? And so for me, I think the question is, have we done enough assays to really understand that? And I think at the moment the answer is no. We've done a significant number of assays where we get very good data from really non-disease relevant models, non-disease relevant cell types. So I think we know that it's definitely possible to get incredibly useful data that's definitely an improvement on anything that we can do computationally from really disease irrelevant contexts.
Like being in a yeast cell.
Like being in a yeast cell, for example. So we've got enough examples where we've proven that. But what we don't know is that true 80% of the time, 50% of the time, 95% of the time? And that's where we need to get more data. And then that will be empirical. And then we'll start to be able to say, okay, so there's these types of genes or these types of functional elements where actually cell context really matters. Now what we understand about a lot of the protein encoding genes is a lot of the damaging mutations are protein destabilizing mutations. Certainly from a perspective of loss of function. And that's going to be proteins destabilizing in most cell contexts, whether it's a yeast or and so we know that anything that's reasonably informative about physiological protein destabilizing should be partially informative. But whether it's fully informative, we won't know. But that's where, fortunately, we've got this benchmarking data. So if we start to see genes where 10% of the variants are misclassified, and we start to understand, well, what is that? Is that because it's part of the protein that interacts with a protein partner that's only present in some cell types? Then we can start to develop predictive models about which of the genes where it really matters to get the right cell type. I think that, in some sense, all bets are off when it comes to non-coding sequences, because we know that non-coding sequences operate in much more cell-specific ways. And so it may well be that the context is much more important as we move into the non-coding space. But that's, again, will be a testable empirical kind of question. Unfortunately, in that space, it's also a challenge for identifying, gold standard benchmarking variation data sets. So I think that's probably the area where I think there's the biggest question mark is in the non-coding space.
Yeah, and I'm really interested in environmental dependencies too. So working on pharmacogenes, how much of the variation we see is substrate dependent? Like, oh boy, that's one of the things that keeps me up at night a little bit is like, we've crafted these assays with these little molecules that are not exactly like the drugs that matter for real clinical implementation. And so we're trying to think of ways to better explore some, more clinically relevant, more medically relevant assays that use the real molecules that matter versus proxy phenotypes.
Okay, cool. In the interest of time, I'll just ask one last question, and this is a very popular question. So Sina asks, she was wondering how you see the role of machine learning and DNA language models in variant effect prediction in general, and whether the Alliance has any interest in this area.
I could start on this if you like,
Yeah,
Maitreya.
go for it.
So yeah, I think this is, machine learning works well in certain kinds of contexts, where you've got, large amounts of data and where you've got problems that don't naturally fit an easily explicit statistical model. And the kinds of MAVE data that we're talking about exactly fit that kind of approach. So the MAVE data, MAVE community is and the MAVE problems are very amenable to machine learning. So I think I see very much a future whereby we'll have very productive feedback loops between data generation, machine learning guiding new data generation. So I think it's critically important that we have the computational kind of firepower within the community and computational creativity in the community that kind of help that fertile kind of interactions kind of as we go forward, because it's if I compare it to something like variant calling from whole genome sequencing, for example, that's somewhere where pretty simple statistical models do a really good job. The data gets you almost all of the way there. Whereas predicting, what function a protein variant has is a much more challenging, much more difficult, kind of multidimensional, non-linear kind of approach. And that's where machine learning, I think, will be really powerful. Well, that's my take on it.
Yeah, and I will just add to that one thing that I'm excited to see is more kind of nuance being brought to these models, more recognition that the whole genome is not uniform and that they're like different genes have different properties. And having all these data sets from all these different genes, I think is bringing a better view of like what matters to a particular protein, and are there context dependencies that we don't understand yet that could help improve the accuracy of these models for all the different genes and all their different contexts, versus kind of trying to understand everything with a more generic approach. So that's what I'm excited to see where things go. I mean, heck, if you can save me doing a bunch of boring experiments, like I would love to be able to just go right to the answer for a lot of things, but we are certainly not there yet, and the experiments are still fun. So